getting on. Because usually the next day it should work. So if it doesn't work tomorrow, then you need to contact someone at registration. Yeah, I mean, I believe you. There's nothing I can do. So yeah, make sure that you get access to Canvas as quickly as possible.
If by any chance, like, I don't even get access to it tomorrow, should I'm still gonna contact you, but how will I go about, like, because I saw discussions tomorrow, and then the homework one, I guess I'll just do it for regular credit, but like the integrity pledge, I know that's pretty important.
So that's what I just told you. Send me an email so I know to expect yours late. Okay. And that will be fine. Right, that will be totally fine. You won't miss anything. Yeah. All right, let's go ahead and get started. Good afternoon, everyone. All right, just a few quick reminder announcements. So please remember to submit your first assignment. It is due in like an hour and a half, right? Most of you have done it, but 20 of you still have not submitted this. I know some people like to procrastinate, but don't forget, do this, okay? This is really essential. Right after class, homework 1 will be posted. This will directly reflect all the classwork we've had up until the end of class today. So do the homework, go to your discussion section. It will be discussed in your discussion section. Go prepared to talk about your answers, things you're confused with, things that you need more help in explaining. And remember, if you do complete it before 10 a.m. tomorrow, submit it. You will get extra credit for that. And even if you don't do it by 10 a.m. tomorrow, as long as you submit it by noon on Tuesday, you can still get a half point of extra credit. Discussions do start tomorrow, so some of you will have your first discussions tomorrow, and I think there's another section that's on Monday. And remember, you want to participate at those discussion sections. That's important also for points for this class. And then just make sure that your iClicker is registered in order to get extra credit points for that. And many of you have registered and have been accumulating extra credit over these past 2 courses. Does anyone have any questions about these announcements or class format? All right, so let's do a little review of what we talked about in class on Tuesday. And so we talked about, we started off by, we needed a definition of a gene. When we think about our DNA and our genes, we need to understand what that means. So what is our definition of a gene? Yes? Yes, exactly. It is a nucleic acid sequence or the DNA sequence that encodes for a functional gene product. Many times those gene products will be proteins, but sometimes it is a non-coding RNA that is indeed still a functional gene product. So there is our definition. And then we looked at sort of an example model of what a gene can look like and the parts that might be needed for a gene. So if it is a protein coding gene, the coding region is what gets translated, tells the sequence of amino acids to be linked together to make the protein. Sometimes within that coding sequence, there are sequences that interrupt the reading frame. What do we call those? Introns, exactly. So the introns are removed, and everything else of the RNA that gets ligated back together are called generally what? The exons, exactly. And so that will include sequences that get transcribed that aren't part of the coding. They aren't translated yet. And if it's before the start codon, it is the 5' untranslated region, 5' UTR. And if it's downstream of the stop codon, it is the 3' untranslated region. We also said many messenger RNAs will have a sequence in the 3' UTR that is called the polyadenylation signal. This tells cellular machinery to come in and put a poly-A tail, a string of A's, onto the end of the 3' UTR RNA sequence. So Upon transcription, which is going to be regulated by promoter sequences, these are what help recruit the polymerase, the RNA polymerase, to a gene so that a pre-RNA can be synthesized. In this case, it is a messenger RNA because it is a messenger for making protein. And so the pre-mRNA will be everything that is initially transcribed. That will include introns. And then that pre-messenger RNA will be a substrate for splicing. Splicing removes those introns, ligates the exons back together. And the polyadenylation machinery will then see this signal in the RNA to add a poly A tail. This then will produce what we call then the mature messenger RNA. This is the messenger RNA that will go out to the cytoplasm and be translated. Any questions about that? So this is one flavor of a gene and the RNA that comes from it, but there's different types of genes and the way they're synthesized. So we looked at one which is a protein coding gene that happened to have an intron in it or multiple introns in it. There are some messenger RNAs that don't have introns. They don't undergo splicing. There are also some that may or may not have introns, but also do not end in a poly A tail signal. They have a different kind of termination at the 3' end. And then there are also RNA genes. So they do not have a coding sequence. They are not translated by the ribosome, but they make a functional non-coding RNA. Some of them have poly A tails. Some of them do not. Some have introns. Some do not have introns. These are all examples of genes. Questions? All right. Oh, did you have a question?
Yeah, sorry, what does that little symbol mean between the coding? Is that just like a splice there, or?
Right here?
Yeah.
That is a symbol for an intron. Okay, so this is the sequence we said it's interrupting the coding sequence, and so it is an intron that would get spliced out. There's also some mRNAs that don't have introns in them. All right, so get out your clickers. Let's answer this question. So, looking at this model up here, which of the following depicts the mature mRNA expressed from the above gene? And here, these are the exon sequences, these are intron sequences, and then these, of course, are UTR sequences, and then there's a polyadenylation signal here.
Is it always going to have the 5' cap and the poly A tail, no matter what, if it's mature or no?
So there are, remember on the last slide, there are some RNAs that don't have a polyadenylation signal.
Okay.
So in those cases, they would not have a poly A tail. If they have the signal, as shown by this PA, then they will get polyadenylated.
So this is the 5' cap, right? Or no? Is that?
So we didn't talk about cap yet, but it would be at the very 5', that very first nucleotide would be attached to a 5' cap.
Okay, so this isn't the same as a cap, though, right?
It is the 5' untranslated region. It's a whole sequence of RNA. The cap is just one nucleotide that gets attached to the 5' end.
Okay, so that's what I was thinking this was, but that's not.
No, and we'll talk more about the process of capping and polyadenylation in a future class. All right, 10 more seconds. Get your answers in. All right. Oh, we finally have a majority liking one of these answers. So everyone, or most people, like answer D. Why do you like answer D? Sure.
We have to take
Yeah, exactly. So when we're talking about the mature messenger RNA, it is an RNA that has gone through the whole process it needs to go through to take out introns, if it has introns. This one depicted has introns. It has a polyadenylation signal here, so it needs to get polyadenylated. These UTRs, are they part of the mature final messenger RNA? Yes, absolutely they are. And in fact, the 5' UTR is really a part of what? Is it an exon? How do we define introns and exons? What are introns? They are in the way, gets removed. Everything else is an exon that gets ligated back together. So are UTRs exons, part of exons? Yes, they are definitely part of exons. Exons are not defined by having coding capacity. We would call these coding exons the way that I have marked it here. But remember, even non-coding RNAs sometimes have introns, and therefore the whole rest of the RNA after the intron removal is the exon. So exon, or the 3' UTR is a part of what? Exon 3. The 5' UTR is a part of exon 1. Does that make sense? All right. And again, we'll go over more the details of splicing in a future lecture where you'll see this even more clearly. But for now, definitely these UTRs are part of this mature messenger RNA. Any questions about that clicker question? All right, so talking a little bit more about what we discussed in class on Tuesday, we talked about all the different types of DNA that are found in our large genome. We indeed talked about different types of protein coding genes. Sometimes they look like they're once in the genome, no really homologous family members, so those would be solitary. Then there were duplicated ones where related family members looked and have slightly different sequences and functions. And then we talked about these tandemly repeated genes, which tended to be the same exact gene repeated often over in a row, and that's because we lead a lot of expression of these types of genes. So these would be like your ribosomal RNAs, your tRNAs, and your histone protein coding genes, which we'll talk more about today. And then finally, we talked about all this repetitive sequence, which is in our genome. Sometimes these repeat sequences, when they are within protein coding genes, can expand. They can change either the sequence of that protein coding gene and cause it to have a disease phenotype, or they can change how that gene is going to be expressed, like if they are found in a UTR or an intron sequence. They can cause that gene to be expressed too highly or too lowly, and that can lead to the disease phenotype. And then the last thing we talked about were the transposons that are found largely throughout our genome. And so we talked about two basic types of transposons. They are defined as either DNA transposons or retrotransposons, which is just another name for RNA transposons. And there are a few differences between these two transposons and how they mobilize within the genome. We saw that DNA transposons use this cut and paste mechanism. What that means is the original DNA sequence, the original DNA transposon, hops out of its original location, and then those two ends will get ligated back together. This is where the transposon originally was. And then the transposon will have an activity to open up DNA at a target site, insert itself, ligate itself back in there. So the DNA transposons jump from one position to another position in the genome. On the other hand, we have RNA transposons or retrotransposons, and they move in a copy and paste mechanism. What this means is that the original sequence, the original retrotransposon sequence, stays. Wherever it was located in the genome, it stays there. But once it gets transcribed by an RNA polymerase and makes an RNA intermediate, that RNA intermediate can then be reverse transcribed. We said reverse transcription means you use an RNA template to make DNA. After reverse transcription, we now have a copy of that RNA transposon that then will insert somewhere else in the genome. So what happens here is we have gone from one copy of this RNA transposon to now two copies, right? Because it has copied itself and put a copy of itself somewhere else. What about the DNA transposon example? Do we end up with two copies? No. It's the same one, but it's just moved to a different position. Any questions about those mechanisms? Yes? Potentially, and depending on what types of transposons you're talking about. And so we'll lead a little bit into that now. Most transposons in our genome are pretty much silenced and not active, but there are, especially our L1 transposons, that are active actually during early embryogenesis, and then they start to get turned on as we age or stress or under cancer conditions in ways that probably are not beneficial for us. So we'll get to that point in just a moment. All right, so let's talk a little bit about the role transposons have played in gene evolution. So remember when we talked about duplication of the globin genes? So we have those multiple globin genes that then have diverged a little bit and have different oxygen-carrying properties. Well, one of the ways that we think that these duplication events have been mediated has actually been through repeat sequences or transposon sequences that flank these globin genes. So these pink boxes on either side here, these are just repeat sequences or a type of transposon sequence. And because this sequence is exactly the same as this sequence, you can get unequal lining up, and you can get unequal crossover. And we saw that as an example of how some of these globin genes got duplicated. Now, if that can happen, we also have recognized now that in some cases we can even create entirely new genes through a process called exon shuffling. And so what that means is if you have two entirely different genes, different chromosomes, but they have repeat sequences in the introns as drawn here. ALU, remember, is a type of sign transposon. It's just a short sequence. But this sequence is exactly the same as this sequence, is the same as this sequence, is the same as this sequence. So if during recombination these two genes happen to get close to each other, you potentially can have a crossover event which will take these first two exons of gene 1, cross over here, pick up exon 2 of gene 2, cross back over, come back up here, and now have the last two exons of gene 1. So you have shuffled the exons, and then you've also exchanged this exon down into this gene. Many cases that will make a non-functional protein and won't do anything, but there are some examples throughout evolution where we do recognize this event has happened and it has given that protein coding gene a new function by putting some new amino acid sequence into it. And much more commonly, one thing that these transposons have done by jumping around our genome is they have changed regulatory regions that control gene expression. So for example, if this is the promoter region of a gene that drives low-level expression of this gene, if you now have a transposon jump into that promoter, it could add a sequence to bring a new transcription factor binding site to increase expression of that gene. It could remove a negative regulatory sequence so that now you'll get higher expression of this gene. And so by changing the way that genes are recognized for transcription, you can change the level of expression or perhaps what tissues the gene is expressed in or in response to different conditions. In my lab, we discovered an element that has jumped around the worm genome and has brought several genes under the heat shock response. So these genes now turn on when the worms are heated up, and that's because a transposon has jumped into their promoters. Of course, there's many bad things that transposons do by jumping around the genome, and we can see evidence of this, of them moving into positions of very important protein coding genes and changing the way that they are expressed, and these can lead to diseases. The examples all shown in this table from your book are actually introns that have jumped into introns. How could a transposon moving into an intron change expression of that gene? Don't introns just get removed and spliced out? Sure. It can change the intron and the way that intron will be recognized for splicing, right? So when we talk about splicing, we'll learn that there are specific factors that recognize these splicing signals, and it's essential to know here's the intron, we need to remove this sequence and put the exons back together. If you change that sequence by putting a transposon in there, you can change how well that gene will be spliced. That could affect the actual sequence that get the mature messenger RNA sequence, which would change the protein sequence. It could change how stable the RNA is, so maybe you have very little mature RNA coming, and therefore you have a lot less of that protein being available. And so all those different things have happened with these different genes that have had transposons move into their introns, and that has caused changes many times in exons being skipped, or sometimes the RNA even being unstable and not expressed, and therefore you don't have mature RNA, so you can't make the protein. Question?
Do both the DNA and the retro RNA type transposition, are they like equally likely to happen, or is there one that's more common?
In the human genome, the L1, line 1 is more common, but it's different in different species, in different organisms. Humans, L1. It would be, so L1 is an RNA transposon, so it would be the copy and paste. And then also in the past 10 or 20 years, we've also started to recognize that transposons can start to become mobilized under certain conditions in different tissues throughout our body. So our cells normally have a lot of defense mechanisms to keep transposons repressed, to make sure that they are not transcribed into the RNA. If you don't transcribe an RNA transposon, it's never going to mobilize, right? There's nothing to reverse transcribe. So if you shut down transcription of a transposon, you don't have to worry about it. And there's a variety of mechanisms that help shut it down. Many of them are at the chromatin level, which we'll talk about later in today, different ways to regulate the availability of genes within different kinds of chromatin. But what we now recognize is there are events like stress conditions, constant stress conditions, being exposed to DNA damage, certain pollutants, environmental factors, and aging just itself, which is kind of a form of stress, actually seems to weaken these defenses, which means these sometimes can start producing RNA, and then they can start making their transposon proteins, and they can start jumping around the genome, which then can lead to genomic instability, which we think could underlie perhaps some aging phenotypes, as well as being involved in cancer. Many tumor cells tend to see higher levels of transposon activity, which we think leads to genome instability. And so because of these problems with transposons moving when they're not supposed to be, there's a lot of interest in trying to understand how transposons, the actual mechanisms they use to mobilize and insert in various places around the genome, and how we might be able to control this for therapeutic uses. And so I posted this review paper on the website. I know at least one of you read it already, so great. So if you're interested in transposons and different ways, different therapies that are being developed to potentially inhibit them from moving, I recommend you read this. Anything you read in there that I haven't talked about in class, you're not going to see on an exam. This is for your own interest. And this is just a figure from that paper that goes into a little bit more detail about how these line 1 or these RNA transposons move. But it's basically what we've talked about. The line 1 is a sequence in the DNA. Even though it's an RNA transposon, it is in your DNA. When it gets transcribed into RNA, that RNA can go out to the cytoplasm, get translated by the cellular machinery into the proteins it's going to need for the RNA to go back into the nucleus, get reverse transcribed into DNA, and then integrate into the genome. Any final questions about transposons? All right, so now let's move on to some new material tonight. So we're going to focus tonight on the structural organization of eukaryotic chromatin and chromosomes. So if you think about it, almost every one of our cells has 46 chromosomes. And the average size of a single chromosome, that DNA, if you stretched it all out, is the size of my pinky. All right? You have 46 of those in every single one of your cells. So how in the world does a cell compact so much DNA into such a small amount of space? And the way it does it is through forming chromatin. So by folding and wrapping that DNA around proteins, and the individual group of proteins we call nucleosomes. And we'll look at how those are composed of specific histones in a few slides. Now, by packing that DNA, we can either pack it very tightly, condense that DNA into very tightly packed chromatin. We call that heterochromatin, and this is usually associated with inactive gene expression, which you can imagine, right? Because if the DNA sequence is tightly packed into chromatin, it's going to be very hard for the transcription factors to get in and find that sequence and transcribe that gene. Most of your transposons are tightly packaged up in heterochromatin. In contrast, genes that you want to express, that you want to have being transcribed, these are wrapped in nucleosomes in a form that we call euchromatin. This is a more open, active form of chromatin. So looking at these two pictures, is this picture in A, do you think this is euchromatin or heterochromatin? Euchromatin, right? We can see sort of the DNA on these little beads here. These are the individual nucleosomes. It's sort of more open, looser. So this is heterochromatin, these like dark, thicker bands here. This is the more packaged, the condensed version of chromatin. And one of the major things that regulates how loosely open or tightly packed a region of DNA is going to be modifications that happen to the histones that are associated with different regions of the DNA. And then we will talk about, I think the first experiment we're going to talk about in this class, it's an experiment called a DNase 1 protection assay. And we can do this assay to simply ask, is a particular region of chromatin we're interested in? Is this line 1 we're interested in, is it open in this aging tissue, or is it in a more condensed version and therefore more likely to be inactive? All right, so this is just the roadmap to remind you. On Tuesday, we talked about the major types of DNA sequences. And then today, we're moving in to see how all that DNA gets packaged into each of our individual cells by wrapping around histones in forms of complexes we call nucleosomes, and then how those get more tightly wound into your chromatin structure. All right, so it's been a long interest to know how is chromatin actually packed inside the nucleus of our cells. And earlier electron micrograph pictures show this very distinct packaging where these dark areas right here, these were the heterochromatin, where the chromatin was more tightly together, and these more white open areas here is euchromatin. More recently, a more refined way to do electron microscopy looking at chromatin in live cells has shown that it's not quite so black and white as this. We see some regions where it's more darkly packed, and that is indeed heterochromatin. And then these sort of looser areas we recognize as euchromatin. And the reality is that chromatin in our cells we think of now more as just sort of a disordered change of nucleosomes that are sort of packed together at different densities. And so when it's looser, sort of sometimes it's called beads on a string, those beads being these nucleosomes, this would be associated with euchromatin or more open and more likely to be active genes, versus when it's more tightly packed together, we call that heterochromatin. All right, so let's define, so what is chromatin? What composes chromatin? All right, so chromatin, at the base, you're going to have DNA. The reason you have chromatin is to wrap up that DNA and pack it in your cells and be able to regulate its availability. And the way the DNA is going to be packaged is by proteins. And the major proteins that are packaging our DNA are proteins that are called histones. There's in general, most of our chromatin is composed of these histones. So H1 stands for histone 1, H2A, histone 2A, histone 2B, histone 3, and histone 4. If these are proteins that are going to be tightly interacting with our DNA, what kind of general charge do you think these proteins contain? Positive. Why? That's where you get the positive. Why do you want to be positive? Right, interacting with the DNA, and your DNA is very negatively charged, right? And then in addition to our chromatin having the histones with the DNA wrapped around it, there's going to be other proteins also associated with the DNA and the histones. There's proteins that help form the structure of the chromatin, scaffold proteins. There's proteins that are going to be acting on the DNA, like transcription factors or replication factors when it's time to copy the DNA. All right, so here's our DNA, and about 147 base pairs will wrap around a complex that we call a nucleosome. And what that nucleosome is composed of is an octamer. Octamer means 8. This is 8 proteins, and those 8 proteins are 2 copies of histone 2A, 2 copies of histone 2B, 2 copies of histone 3, and 2 copies of histone 4. And so that is going to be the nucleosome around which the DNA will initially wrap. And then in these linker regions where there's a space and it's not wrapped around a histone octamer, that's where your histone 1 is going to bind to the linker regions of the DNA that's between those nucleosomes. Question?
Does the DNA wrap around the nucleosome or the histone?
So our definition of nucleosome is the histone octamer. Okay. So the nucleosome is the 8 histone proteins that are 2 copies of these 4 different ones.
So when the DNA wraps on the histone,
So the nucleosome will be the 8 proteins together, and then the DNA will wrap around that. And so actually the next picture might help you see that even better. So here is actually a structural picture of what that looks like. The 8 different histone proteins are shown here in the sort of ribbon drawings in sort of the center, and wrapped around the core of those proteins, wrapped around that octamer in white here, is the DNA. So this is the double-stranded DNA that is wrapped around the nucleosome. The nucleosome is those 8 histone proteins. All right, and so sometimes we'll be talking about histone tails. By tails, we just mean the termini of the protein, the beginning, the N-terminus, or the end, the C-terminus. And those are the regions that sort of stick out here. So these dashed lines, they're kind of flexible, and that's why they're drawn with dashed lines here. And so as you can see in the drawing here, what do you think the histone tails interact with? The DNA, right. You can see it with the DNA here. And also because they're sort of on the external part of the nucleosome, they're also available to interact with other proteins. So there will be proteins that will recognize these histone tails, especially modified versions of these tails. We'll see different proteins will associate with the histone depending on what modification they have. I think we already said which positively charged amino acids will facilitate this interaction, which are our 2 major positive amino acids. Lysine and arginine, right. And then, does anybody know examples of any kinds of modifications that occur on histones? Sure. Methylation is one. Sure. Acetylation. Anyone heard of any others? Deacetylation would, I guess, kind of the absence of formally being acetylated. Phosphorylation is another one. Yes. And then the other one is ubiquitinylation. Some of you may have heard of ubiquitinylation in regard to degrading proteins, but sometimes if you just have a single ubiquitin, it is not recognized for degradation. It is recognized as a modification for other proteins to bind. So here are examples of common histone modifications. And so what you're looking at here are 5 different histones, right? So this is histone 2A. This is the serine, the amino acid one. Is this the N-terminus or the C-terminus of the protein? At the first position. It's the N-terminus, exactly. So the beginning of the protein is the N-terminus. The end of the protein is the C-terminus. And so what's being depicted here is at this S, or serine 1, it can be phosphorylated. It's not always phosphorylated. It's a very regulated event. And then there's a variety of other positions shown here. Histone 3 is the histone we'll talk about most. It is the most consistently associated with certain being active or being in repressed chromatin based on a few key positions. So if we look at position 9 here, we see a K. What is K? Which amino acid? Lysine, exactly. So we have a lysine here, and we see Me stands for methylation, and we see AC stands for acetylation. What that means is this lysine 9 position can be methylated or can be acetylated, but it can never be both at the same time, and we'll see why that is in a moment. And depending on whether it's acetylated or whether it's methylated has a huge difference in the type of chromatin that it will be in. Don't memorize these modifications and their positions or anything like that. The ones that we'll talk about over and over again, those are the key ones that I want you to keep in mind. So let's look at what that means to be acetylated versus methylated. So here is a side chain of an amino acid. Which amino acid is this? Lysine, exactly. All right, so here we have a lysine. And what do we notice? We said lysine is positively charged. So here in the nitrogen group, this is the positive charge. And we said that lysine can undergo 2 different types of modifications. So green arrow, which modification is this? What kind of group is that? Acetylation, exactly. And this one is methylation, right. So let's look. What happens here when our lysine gets acetylated? Well, we add this group onto it, but what else did we notice changes? The positive charge, right? We lose the positive charge. All right, we said when we're positively charged, that makes it more favorable for interacting with DNA. If we lose the positive charge, you're going to have looser interactions with DNA, right? So typically, at least for the modified positions we're going to talk about, if you are acetylated, you're going to be in a more open conformation. You're going to be more loosely associated with the DNA because of this charge change. All right, here we said we have methylation. What has happened to the positive charge here? It's maintained, right? And it turns out that this can actually have a mono or a single methylation, or you could have another methylation, and then it would be a dimethylation, or even at this position in addition, and then you would have trimethylation. In any one of those cases, mono, di, or tri, you still keep the positive charge onto it. So with the positive charge, you stay tightly associated with DNA. So methylation is going, at least in the cases we're going to talk about, is going to be more associated with condensed or tighter or more heterochromatin situations. All right, does anybody know the general name of the enzyme that would acetylate a histone? If I tell you the abbreviation is HAT, can anyone take a wild guess? Yes? Yeah, histone acetyltransferase. Based on that, anyone want to guess what the general name for the methylation one is? Histone methyltransferase, exactly. So the enzymes that take histone positions and acetylate them are called histone acetyltransferases, sometimes abbreviated as HAT. They add a methyl group. They are generally called HMTs, or histone methyltransferases. All right, so here's some examples of what I was talking about. So here we're just looking at histone H3, and this is pretty much the only one we're going to talk about when we're talking about modifications, but please appreciate there's many modifications on all of the different histones, and they all have important functions. But these are some of the best studied. So it is well established that when the 9 lysine position of histone 3 is methylated, that is typically associated with heterochromatin, or tight packaging of the DNA that is associated with this type of modified histone. Also, the 27 position is typically methylated to be associated with condensed chromatin. On the other hand, our Euchromatin or active chromatin, we would have acetylation on the lysine 9 position, and often at a variety of other positions. Of course, because biology can never be straightforward, there's also a methyl on the specific lysine 4 position that is actually more associated with euchromatin. This has to do with the proteins that recognize that modification to help open it up into a looser form. All right, so what we often say is that depending on which modification and what groups of modifications the histones have, they are read. They are read by the cellular machinery. How is the histone code read? What does that mean? Any ideas? We said the modification can affect how well it associates with the DNA, but what else do those modifications do? There's different proteins that will bind to that histone only when it's methylated. In another set, only when it's acetylated. In another set, only when it's not modified. So there are specific proteins that read this code. And we'll talk about one example here. So there is a protein that is called HP1 that promotes heterochromatin formation. HP1 stands for heterochromatin protein 1. It's an example of something that is very nicely named for what we know about it. All right, so how does HP1 read the code and promote heterochromatin formation? So when the lysine 9 on histone 3, so remember K is lysine, the 9 after it means the 9 position, the 9th amino acid, and H3 means histone 3. So when the histone methyltransferase for that specific position puts, in this case, trimethylation on the position, this is recognized by HP1. HP1 will only bind histone 3 when it has either 2 or 3 methyl groups on that lysine 9. So if you start to get that modification on your lysine 9 and histone 3, HP1, the protein, reads that and binds directly to that modified histone. HP1 proteins themselves not only bind to the histone, but they like to bind to each other. They stick to each other, and that's what we call oligomerization. What that means is all these HP1s that are bound along here then all start sticking together, and so it starts pulling all these nucleosomes together. You can see how this is starting to look like heterochromatin, right? It's really compacting the chromatin. But then HP1 actually has a third trick that it uses. It not only binds to the methylated histone 3 lysine 9 position and causes oligomerization, but the protein itself actually recruits the histone methyltransferase that puts the methyl group on lysine 9. So what it's essentially doing is it's bringing in the enzyme to make more substrates for itself to bind to. So HP1 is binding to the enzyme to add that modification onto nearby histones. And so as you can imagine, this is how you can spread heterochromatin along a region of chromatin. You're getting HP1 binding, oligomerizing, and then recruiting the enzyme to make more binding sites for itself. Now, eventually, this needs to stop. You don't want your whole chromosome to turn into heterochromatin. And so one of the ways that this process stops is there are what are called in our DNA sequences boundary elements. It's a good name for it. There's a boundary. You stop this spread. And a boundary, another word for a boundary element is insulator. Maybe some of you have heard of that. It insulates you from this type of chromatin from spreading. And this is just going to be a sequence of DNA that prevents the spreading of the chromatin state. And one of the ways it can do that is by being bound by a protein that's not a histone. So if you have a transcription factor or scaffold protein that isn't histone 3, there's no substrate for this methyltransferase to put a methyl group on. There's nothing for HP1 to bind anymore. And so that's how you can stop this spreading. Now, if we have proteins that read this type of modification to promote heterochromatin, you can imagine there's also going to be proteins that instead promote euchromatin. And so there are specific proteins. One example is a protein called TF2D. You'll hear about this protein again. It's a factor involved in a basic transcription by RNA polymerase II. But it actually recognizes the acetylated form of histone 3 on lysine 9, binds directly to that, and then promotes this opening or looser chromatin. Again, the way that euchromatin doesn't spread throughout the whole chromatin is that eventually there's a boundary element so that there can be this regulation maintained. Any questions about that? All right, the last thing I want you to keep in mind when we're talking about chromatin is that there's a lot of structure involved in chromatin, including the ability of it to loop out. And so what you're looking at here, so this would be all the nucleosomes here together, but these nucleosomes can fold back, so you can have sequences that are very distant in 2 dimensions be brought very close together in 3 dimensions. And it's important to have this concept in mind when we talk about the process of transcription, because we're going to learn that there can be sequence, there can be promoter elements, regulatory regions called enhancers that can work from very far distances, hundreds of thousands of nucleotides away from the gene that they are going to regulate transcription. And this is easy to recognize if you can imagine that even though this element A might be 100,000 nucleotides away from element B, it can still regulate the transcription of this gene because in 3 dimension, this sequence will be very close to the start site of transcription for this gene. Any questions? All right, so now let's talk about an experiment that we can use in the laboratory to ask, is a certain region of DNA, is a certain gene we're interested in a particular cell type, under a particular condition, in open chromatin and therefore more likely to be expressed, to be transcribed, or is it tightly condensed in heterochromatin? And the name of this experiment is DNase 1 protection assay. Because essentially what we're asking is the sequence we're interested in this example globin, is it protected by being in very tight chromatin, or is it open and they're susceptible to degradation by DNase? Remember, DNase is an enzyme that chops up DNA. But it can only do that if it can get to the DNA. The DNA is very tightly packed in all these proteins. It's going to be protected. It's not going to be chopped up by this DNase enzyme. So here we have the same gene we're looking at in 2 different cell types. So we have a mature erythroblast. What are erythroblasts? What type of cell? What are they? I'll give you a hint. They do express globin. Yeah. Yeah, red blood cell. Yeah, red blood cells. And that's being compared to MSB. This is just a type of undifferentiated cell type. So this is not a red blood cell. You would not be making globin in it. And so you can already predict that in our red blood cell, we would expect globin to be in decondensed or Euchromatin, so we can transcribe the globin gene and make lots of globin protein. Whereas in another cell type where you don't ever want to transcribe this gene, let's pack it up in heterochromatin. All right, so how does this experiment work? Well, the very first thing you do, since you're asking, what is the structure of the chromatin around my gene of interest in these two different cell types, is you're going to isolate the nuclei. So you're going to get the nuclei from those cells because that's where your chromatin is, right? And then you are going to add to the chromatin, these two different samples, DNase 1. What's DNase going to do? It's going to go up and it's going to chop up all the DNA that it can find. So anywhere where you have loosely packed DNA, DNase is going to go in and degrade, right? If it is tightly packed in heterochromatin, DNase isn't going to be able to access it. It is going to be protected, hence the name DNase protection assay. All right, so after we do our DNase treatment, what we would be left with at this region of the genome is essentially no DNA. It got all degraded. But here, this region is not nicely packed, so that would still be intact. All right, how are we going to detect whether our gene is intact or has been degraded? The next step is you purify the DNA. So you get rid of all the protein, all the other components that were in those nuclei. And then, because we know the sequence of the human genome, we can choose restriction enzyme sites to cut out a region around our gene of interest that will give us a size of DNA that we can run into an agarose gel. And you'll see why that's important in a moment. So the globin gene has BAMH1 sites. BAMH, what is a restriction enzyme? What does it do? It cuts DNA, but only at specific sequences. And so that's why it's quite different from DNase. DNase 1 goes around and cuts up DNA, doesn't care what the sequence is, pretty much. BAMH1 is going to look for a 6 nucleotide sequence, and only when it sees that is it going to specifically cut. And so it turns out the globin gene has these 2 BAMH1 sites, which means when you treat your purified DNA with BAMH1, you are going to cut out a nice sized piece of DNA that has our gene sequence of interest. The enzyme's also going to cut millions of other places in your total genome, right? We still have all of the DNA that was in the nuclei in that cell. And so what you're going to have then are millions and millions of pieces of DNA within your test tube. And so then you want to be able to detect, is my globin gene sequence, was it protected from DNase or not? And so to do that, we're going to design what we call a probe. And the probe is going to be designed to hybridize is another word for base pair, to your specific sequence of interest. So we want to ask, is the globin gene protected? So we're just going to design a probe that can base pair to a sequence within the globin gene. So if I tell you that the beginning of the globin gene begins with this sequence here, what sequence would you use for your probe? Let's say starting here, 5 prime. What is the first 3 nucleotides of sequence of your design probe? A, C, G. Exactly. You just use your base pairing rules. So we want our probe to hybridize or base pair to a sequence within the gene. Do I need to make a probe that is the entire 4.6 base pairs of DNA? No. It just needs to be long enough so that it will be specific and only base pair to globin and not somewhere else in the genome. Typically, you need to be at least 20 to 30 nucleotides long to get that kind of specificity within our genome. Is that your question? Okay, great. All right, so great. So we've made a sequence of DNA, and you can synthesize these in the lab. You can order them. Basically, you will get a sequence that will base pair to your region of interest. But now we're going to need to be able to detect that hybridization happened. Can anybody name a way that we can detect our probe? Yes? Yeah, yes, you can add fluorescence to it, or radioactivity is another way we commonly use in the lab. So, to design our probe, there's two things you need to keep in mind. One, your probe needs a base pair to a sequence within the gene you're interested in. That's how you're going to specifically look, was my globin gene protected? And then I need a way to be able to visualize this or detect it in the laboratory. And you can do that by making sure that your probe either has some radioactivity in it or a fluorescent label. All right, so what are we going to use this probe in? We're going to use it in a type of experiment that's called a southern hybridization experiment. And this simply means that we are using a probe to detect DNA of interest. All right, so from our DNase protection assay, remember what we are left with now is we have our tube. We have all the DNA that was not protected degraded, and then we use our restriction enzyme to cut up everything else into smaller fragments of DNA. We're going to then take that total DNA sample, and we are going to run it using electrophoresis through a gel. What this is going to do is it's going to separate the DNA based on size. Do bigger pieces of DNA run towards the top, or do they run all the way to the bottom faster? Top, right? If you're big, you slowly migrate. If you're small, you move much faster through it. So this type of gel is going to allow the DNA to separate based on size. All right, has anyone run a gel before? You know what gel material's like? It's pretty flimsy, right? It's kind of like Jell-O. So the next step you need to do is you want this DNA to get then transferred onto a more stable substrate. And so nitrocellulose is a type of substance. It kind of looks like a piece of paper. It's a little bit more special than a piece of paper. But what you can do is then you can use capillary action to get the DNA that was in the gel to move right into that nitrocellulose. So the DNA has been transferred onto the nitrocellulose in exactly the same pattern it was on the gel. So at the top, we still have our big pieces of DNA, and the bottom, we have our small. We then use either chemicals or UV light to fix the DNA so it is stably attached to that nitrocellulose. And now we're finally going to get to use our probe. We're going to do the hybridization step. You just mix your probe with a solution and let it float over your nitrocellulose at an appropriate temperature. And that probe is only going to stick to the DNA to which it can base pair to. And so if your globin DNA band is there, and there's a lot of it, you're going to have a lot of probes sticking to that particular position. And so at the final step, you're going to detect it. And so if your probe was fluorescent or it was radioactive, you'll use a mechanism to detect and ask, do I see a lot of probe there? If I see a lot of probe there, then that means that sequence of DNA was protected. If I get barely any detection of that band, then it was probably all degraded by the DNase, and it was not protected. So let's review these steps and think about how we get a correlation between activity and chromatin condensation. So we said that in our red blood cells, we expected the globin gene to be in loose, Euchromatin, and therefore, when we do our DNA step, it's going to get all degraded. On the other hand, these undifferentiated cells, we expect not to be making globin. This globin gene is going to be tightly wound up in heterochromatin. It's going to be protected. DNase is not going to be able to degrade it. And so then after we do our DNase treatment, we'll extract the DNA, we digest it, and that's just to get the nice sizes of DNA that will actually run into the gel. Otherwise, if this whole region, if this was like 1 million base pairs of DNA that's all wrapped up and protected, is that going to run into our agarose gel? No, it's going to be all stuck at the top. Your experiment isn't going to work. All right, and so then we do the southern blot, right? So we run the DNA based on size through the gel. Gel is flimsy, so we need to transfer it onto nitrocellulose. We fix it so it's stably in that pattern in the nitrocellulose. We add our probe. The probe will base pair to any sequences that match it. And then we detect the probe. And here's an example of what those results could look like. So What they're looking at right here in the first example, and this is a very important control, is here's our erythroblasts, our red blood cells. So these were the nuclei that they are starting with. But in this situation, they are using no DNase. Why would it be important to do a control with no DNase? Yes?
You can see all the DNA that you're working with, and then kind of pick out what you need.
So you can make sure that the band that you're interested was there, and you could detect it if it didn't get degraded by DNase. So this lane had everything happen the same. The restriction enzymes were used. It's just that no DNase was added to that tube, and we see a lot of that band being detected, which means there was probe bound to it. But then let's see what happens in these erythroblasts as we start to add increasing amounts of DNase. What happens to the detection of that band? It goes away, right? That means DNase chewed it up. And you can then compare that to our other cell types. So if you're at the highest concentration of DNase, either in the undifferentiated cells versus these red blood cells, we detect none of that band in the red blood cells. But in the undifferentiated cells, even at this highest concentration, what do we see? We see a band there. So what can we conclude from this about the chromatin state in these two cell types? Sure. Yeah, why is it condensed? Yeah, which means that we poured in a ton of DNase and it was protected. To be protected, you must have been in very tightly wound chromatin to not get chopped up by all this DNase. So if we see a very strong band, that means that there was a lot of that DNA sequence for the probe to bind to, so it was therefore protected. It was in more heterochromatin, a more condensed type of chromatin. In contrast to our sample where we use the same concentration of DNase, and wow, we see nothing. That must have been very loose, unprotected globin DNA in that sample. Question? Depends what cell you're differentiating into, right? So if you are differentiating into a red blood cell, like erythroblasts, then yeah, you want to turn on your globin gene. So there will be regulatory processes that will decondense that chromatin and have transcription factors available to make it. But your brain cells aren't making globin, so it's still going to be condensed in that cell type. So it depends on what type of cell you're becoming. Question?
Why doesn't DNase over time still break down the DNA? Like, are the boundary factors stopping the DNase, or is it like time control?
So this is in vitro, right? So in this, yeah. So maybe what you're asking is, if I kept adding even higher and higher amounts of DNase, would just like some normal motion in my test tube lead to degradation? Yeah, probably. Yeah, eventually you could degrade this if you gave it enough time, threw in enough DNase. But what you want to always be doing is comparing perhaps two different cell types, two different conditions, and saying one compared to the other, am I seeing a difference here? And therefore one is more compact and one is more open. All right, let's do a clicker question to see how well you guys understand a DNase protection assay. So above are the results of a DNase 1 protection assay to analyze the chromatin state of gene X before, the minus, and after, plus, drug treatment in various tissue types. The data support which of the following possibilities? Oops, sorry. That's not what I want to do. You can talk to each other.
She's like really sick, and like I can feel myself getting really sick. Like, is it okay if I like leave? Because like I can feel myself like getting sick.
Yeah. Leave or take it or sit somewhere else. Yeah, that's. There's some seats over there. I'm sure. Yeah, actually there is a seat because there's only the only. Yeah, go ahead. Yeah. I'm sorry about that. Yeah. No. Yeah. 10 more seconds. All right, final answers. All right, again, we have a good majority voting for A. All right, who wants to explain their answer? You have good company if you chose A, I guess. Sure.
I can say that it's A because, first of all, it's not, C is incorrect. The spleen didn't show any signals. D is also incorrect because it showed different activation in the heart and liver cells. And then I can say that it's A because after you, when there's the positive, when you have the drug inside of it, and you have the DNase, there's no line, which means the DNase was able to break down the gene X, which means that when the drug is added to the heart, it activates the gene that the DNase breaks down.
Good. All right, so let's go through it. So A is indeed the correct answer, and let's go through all of these, though. So gene X is activated upon drug treatment in heart. So how did he get to that conclusion? Well, he went up here, and he's just looking in the condition where we've actually added DNase. So to know if it is protected by DNase, that is the situation you want to look at here. And it's either with or without the drug. And what you can see is there's a big difference. There's a whole lot of probe binding when there is no drug, and much, much less when there is drug. So what that means is there must be much less of gene X available for binding to the probe because it got degraded by DNase. It was no longer protected. And when it's not protected, it's usually in a more open or a Euchromatin state. And we said that that is associated with being active. And so that the data we have here is supporting this option A. Does everybody see that? All right, let's go through the next one. So gene X is active in the presence or absence of the drug in spleen cells. All right, so let's go. Ooh, all right, what's going on here? Ooh, what's going on here? What happened with our spleen? Can we conclude this about the spleen? No, why not? Our control didn't work, right? If we don't add any DNase, we should see this band, right? What are some of the reasons why we might not see the band in the control? Yes? Yeah, maybe your concentration wasn't right in your cells. It was way, way, way more dilute.
You didn't use the right probe for the spleen?
So on this gel, you'd have the same probe. This would all be on the same gel. So we know since it's lighting up over here, probably our probe is good. What sadly happens many times when we try to isolate DNA or are doing an experiment, some of you who worked in the lab, yeah, it just gets degraded for your pipette was dirty or reasons that have nothing to do with the experiment at all, and it's very sad. But this is why it's very important to do the control. And so for the spleen, we can't make any conclusions at all. We do not have a good sample coming from the spleen because the control didn't work. So we don't know what's going on in the spleen. Does that make sense to people? Yeah. Maybe you even forgot to load samples. You know, things can happen, but there's clearly not the right DNA in our control lanes here. C says the drug affects the chromatin state of gene X in all tissues assayed. Well, first of all, we can't even make any conclusions about spleen, so that kind of rules it out. But what else do we notice up here? The brain, right. So the brain, minus plus drug, anything changing to the chromatin? No. So this gene is the same gene X, but it's in a different cell type. In this different cell type, there's no response to the drug. And then D says the response of gene X is the same in heart and liver cells. So we said in heart, when we added the drug, well, we had less protection. Looks like it became open. Is that what's happening in the liver? It's the opposite, right? So what is happening in the liver when we add the drug? Yeah, it's probably getting condensed and deactivated and less expression of gene X in the liver. And so a single drug can certainly have different effects on the same gene in different tissue types. And then, yeah, definitely not all of the above. So A was the correct answer. Most of you got that. Does these explanations all make sense? All right, so the final thing we'll talk about tonight is the actual morphology and functional elements that are needed in a eukaryotic chromosome. We'll define what we mean by karyotype, and then talk about the 3 functional elements that are needed for a chromosome to replicate and to be inherited into daughter cells. So karyotype simply means the number, shape, size of metaphase chromosomes in a species. For example, here we have a human karyotype. So when we are looking at this fully replicated chromosome, it's being held together. The 2 chromatids are being held together. What is this sequence element in the middle that's holding them together? What is that called? Centromere, exactly. And what are these sequences at the very ends of our linear chromosomes? What are those called? Telomeres, exactly. So those are DNA elements. When I say element, I mean a sequence. It is a sequence. It is a part of the chromosome that is necessary. And in fact, there's one more element that is essential for chromosomes to be copied and inherited. And so there's 3 main things that you need to be a chromosome to be copied and also to be inherited. Someone want to name one of those things? Sure. Well, so maybe you're talking about the centromere. So the region, the sequence element that holds the 2 newly replicated DNA chromosomes together. And what is the job of the centromere? Equal division, exactly. To allow equal division into the daughter cells. So yes, centromere is one element we need. What else do we need? Telomere, exactly. And what do telomeres do? They protect the DNA, and we'll talk more about telomeres in class next week. All right, so we've figured out how we can protect our DNA, how the newly replicated can move into the new cells. We need one more element for the chromosome to actually be copied and inherited. I think I heard it. A replication origin, exactly. So These are going to be sequences that are recognized by the replication machinery in order to start unwinding the DNA and copying it. And that will be the subject of class next Tuesday, how DNA replication occurs. All right, so once we know that we need those three things, would we know how to make an artificial chromosome? What would we need on that chromosome? Those three things, exactly. We need something to tell, hey, copy me, an origin of replication. We would need a telomere, and we would need centromeres. And then, this has actually been made. They're first made in yeast. These are called yeast artificial chromosomes. These can be hundreds of thousands of nucleotides long. They are like a mini chromosome. And in addition to these sequences that are necessary for copying the DNA and for being faithfully transmitted in a progeny, you can pack in lots and lots of DNA sequence of interest, maybe a gene that you want to express highly in that organism, and then they will faithfully replicate and segregate. Based on what we've learned about making yaks in yeast, we now can make hacks. Any guess what a hack is? A human artificial chromosome, right. And so this is very exciting, and there's a potential for using hacks for gene therapy. One of the first examples I'm showing you here, this little pink dot here and this little pink dot here, these are human artificial chromosomes that is successfully replicated and divided into the daughter cells. So they must have had telomeres, centromeres, and an origin of replication. And this is important because there's some human genes we'd like to fix, like the dystrophin gene that's involved in muscular dystrophy. That gene is 1 million base pairs long. You're not going to package that in your typical viral vectors for gene therapy. But if you can get it on one of these human artificial chromosomes, perhaps this might be a way of replacing that gene to patients who have that. And in fact, just a little over a year ago, this paper came out that presented a brand new way of very efficiently making very precise human artificial chromosomes that are millions of base pairs long. And we can do this in the laboratory very efficiently, but like many of the things we're able to do in the lab, getting it into patients and into the cells that needed is still, of course, a very big challenge. All right, that's all we have for tonight. So the last remaining of you, please submit your pledges. Enjoy the homework. Hi.
Like the yeast and the origin of the replication, what's the same thing that all chromosomes will have to have the origin of
Yes. In order to be copied, they will need to recruit the replication.
Okay, thank you.
Sure.
Oh, my name is in like right there.
Oh yeah, then that's fine. Yeah, that's fine.
Question, because we went through all of the amino acids and structures, but I didn't learn that because I was like 41. Is it something we need to know and study in chemistry?
You do not, I will not ask you to draw any amino acids, but any amino acids that I talk about, like you should have a general idea about them, like
About their structure?
lysine is positively charged, what an acetyl group looks like, what a methyl group looks like.
Because the groups I get, because I'm 41, I read like a lot of that.
Yeah.
But in terms of the actual like amino acids or their identities, like that, is this something I need to study on the side?
If you are having trouble answering any of the homework questions or anything, maybe, but I would say it's probably not going to be a big problem. Sure.
So isn't octamer a nucleosome, but a nucleosome is not always going to be an octamer, or are those kind of interchangeable?
The nucleosomes we're going to talk about are always going to be octamers. They're always going to have those 8 core histone proteins in there, but there can be different versions of it for our purposes.
Just kind of leave it for now.
Mm-hmm.
Okay, and then so for the HP1, so they add to the K9, right? And then when it's methylated, but then is there like is this one special? Does it have like any special like transcription factors or like something calling it to start the kind of chain with like these transcription?
It has a domain that can only only bind to the methylated version of lysine 9. And so when that happens, that domain will bind to it. And then a different domain of the protein will stick to other HP1s. And then a different domain of the protein is going to recruit that methyltransferase.
So then are these the same as each other?
Yes.
They are?
Yeah,
Okay.
so all these are the same exact protein binding the same exact substrate. And each one can recruit the methyltransferase.
Okay, so which one, why is this one special if it like starts in? Like what makes that special? Just kind of like something gets turned on or something?
I mean, you know, in reality, if there's a region that has that methyl group, all these HP1s are going to just start binding.
Okay. And then, so with human development, because there is like this, we see that the DNase didn't like chop it all up. Does that mean like in human development, like most of the DNA is going to be like condensed, but then at the same time, like you won't be able to have transcription, right?
So it's going to be different at different times of development. So any genes you don't need to turn on, they can be more tightly wrapped than genes you do want to turn on. And the way we say it, I mean, I'm saying it is like an all or nothing. You're tightly packed, you're like loosely packed, but it's more like a continuum of that. Because you can have high expression, medium expression, low expression, no expression. And there's lots of regulatory factors during development that control that.
In unison with other parts, right?
Yeah, I mean, yes.
Okay, and then last one, so then I chose C, so like how come this wouldn't, because what threw me off is like heart and then I was thinking the liver, but does that mean that the liver is like restricted by gene X in a way because it became like, so there was less DNase that ate this up,
right? So because you have most of it not protected when you don't have the drug, what does that mean about gene X in the liver?
That it becomes unprotected, right? No, it becomes protected because there's more that shows up.
When you add the drug, but it means without the drug, this gene's probably being expressed, right? Gene X is like not protected, it's probably open, but now you add the drug and all of a sudden it's more protected, so it probably got more condensed because of the action of the drug.
Okay, so activated means that it's being like opened up, right? So it's not being...
So open, Euchromatin is going to be more associated with active. And you can think about that. If you're open, transcription factors can see you, can come in and transcribe. If you're all tied up in chromatin, it's going to be really hard to recruit transcription factors.
Okay, so this means most likely it's Euchromatin. Like this.
Yes.
Okay, all right, thank you.
Sure.
Question, how do they have time finding the textbook? Do you by any chance like have a copy or something? Because like I went to the library and they don't have it. And they were
I think there's an online version, right?
I couldn't find it, literally. It was like a sketchy website.
Yeah.
Right, wait. Is it there? Oh, awesome.
Yeah, hopefully you can find it, because honestly, I don't know. Yeah, it should be available. Good luck.
Registered my remote and everything now, but for some reason the app is telling me that it's not finding it, and I already registered it.
Send me an email with all your details, like the serial number and all of that, and I'll look into it and see if it's in my gradebook or what's
Alright, thank you.
Professor, I just wanted to ask, can transposons jump to different chromosome pairs?
Sure.
Okay.
Yeah.
And then you're saying humans have L1 transposon, so L1 I'm assuming refers to L1.
L1 is line 1. L1 is just a short name for lines, line ones.
And then my last question, when we're doing the DNA ACE1, how do we make sure that the length of the gene that we're having is stuck at that 4 million kilo base pairs?
So we know the whole sequence of the human genome, and so if this is a gene I'm interested in, you can go in and look and say, where are some efficient restriction enzymes that will cut out like a 4 kb, 2 kb, 1 kb, whatever, something that'll run into the gene. So you have a lot of flexibility there.
And if it's expressed, that means the DNA is cut it down.
Yeah.
It goes everywhere. We can't really
It's chopped up into little pieces. Okay. There's nothing for the probe to stick to.
Okay, thank you.
Sure. No, as long as that's why I want you to email me, so that I will know. Yeah, no problem.